Welcome to another episode of The Cutting Edge, where we explore groundbreaking scientific discoveries and their implications for the future of medicine and healthcare. Today, we delve into a critical area of research that examines the impact of the anesthetic sevoflurane on brain development. Specifically, we will explore how sevoflurane exposure during early development can lead to neurotoxicity, as revealed through the dysregulation of microRNA, miRNA, or mRNA signaling networks. Background on sevoflurane and neurotoxicity. Sevoflurane is a widely used inhaled anesthetic known for its rapid onset and minimal cardiopulmonary depression, making it a preferred choice in both pediatric and adult surgeries. However, emerging research indicates that sevoflurane may have neurotoxic effects, particularly in the developing brain. This raises concerns about its safety in young children and infants, who are most vulnerable to its potential long-term cognitive impacts. The study, unraveling the signaling network between dysregulated microRNA and mRNA expression in sevoflurane-induced developmental neurotoxicity. This study was conducted by Yo-Yan Wang, Z-Men, Xiaodong Huang, Xiaozhao Hu, Weilong Wang, Zhizhao, and Zhenfeng Zhao. They investigate the effects of sevoflurane on the hippocampus of developing rats. The research aims to uncover the mechanisms behind sevoflurane-induced neurotoxicity by analyzing changes in miRNA and mRNA expressions and their interactions. Methodology. The animal studies. Rat pups postnatal day 7 were exposed to 3.4% sevoflurane for 4 hours. Hippocampal tissues were collected for analysis. Analytical techniques. Western blot and immunohistochemistry to detect neuropoptosis. High throughput sequencing to analyze variances in miRNA and mRNA expression levels. Morris water maze to assess long-term memory in rats exposed to sevoflurane after 8 weeks. Key findings: neuropoptosis and cognitive impairment. Sevoflurane exposure led to increased expression of apoptosis-related proteins, BAX, cleaved and caspase-3, and decreased expression of BCL2 in the hippocampus. Rats exhibited significant long-term memory impairment, as evidenced by the Morris water maze test. Dysregulated miRNA and mRNA. The study identified 5 dysregulated miRNAs and 306 dysregulated mRNAs in the hippocampus post-sevoflurane exposure. Dysregulated miRNA and mRNA interactions were associated with pathways involved in neurodevelopmental disorders and axonogenesis. miRNA and mRNA interaction networks. Bioinformatic analysis revealed that MIR130B5P and MIR449C5P were critical nodes in the interaction networks, impacting various target mRNAs. These interactions are implicated in the regulation of neuron differentiation, axonogenesis, and neurodegenerative processes. Pathway analysis. Go and Cake pathway analysis identifies several pathways affected by sevoflurane, including neuroactive ligand receptors and interaction, cytokine receptor interaction, and metabolic pathways. The study provides compelling evidence that sevoflurane can induce significant neurotoxic effects during early brain development. The dysregulation of miRNA and mRNA networks appears to play a crucial role in these adverse outcomes. Understanding these interactions can help in developing strategies to mitigate the neurotoxic effects of anesthesics and improve safety protocols, especially for vulnerable populations like infants and young children. Implications for future research. Future research focus on further elucidating the specific pathways and mechanisms involved in sevoflurane-induced neurotoxicity. investigating potential protective strategies or alternative anesthesics that minimize neurotoxic traits or risks, expanding studies to human models to validate findings and ensure clinical relevance. The insights gained from the study highlight the importance of carefully considering the use of sevoflurane in pediatric anesthesia. By understanding the underlying mechanisms of its neurotoxic effects, we can work towards a safer medical practice that protects the developing brain. Thank you for joining us on this episode of The Cutting Edge. Stay tuned for more explorations into the frontiers of science and medicine.